Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on November 6th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 66th episode of Matters Microbial. I am grateful to every listener and viewer of this podcast, and thank you all for sharing the word. First, the lapel pins I am wearing today of all days. On the right, I promote this podcast, and on the left, a reminder from my wife, Dr. Jennifer Quinn, that we all need to manifest the future in which we would like to be part. And this is actually a big deal to me as retirement approaches. This week in Bio 350, my Micronauts discuss the identity of their water bottle buddies, bacteria living in their reusable water bottles. I'm asking them to create a one-page summary of their buddies, and there are often surprises, like this particular water bottle buddy that appears to be closely related to the radiation-resistant Deinococcus radiodurans. What the microbe? One of the concepts nearest and dearest to my microbial heart is phenotypic instability. That is, how some genes are not stably expressed by microbes, especially microbes freshly isolated from nature. This variation can be advantageous to the organism, but most microbiologists would prefer phenotypes that do not change. The bacterium Serratia marcescens makes a red pigment called prodigiosin. Some experts have shown that pigment to be toxic to other microbes. Regardless, here is an image of the colonies to be found using a domesticated laboratory strain of Serratia. All of the colonies are brick red. But note this undomesticated strain of Serratia. You can see the individual colonies express different amounts of the pigment and sometimes form fascinating and unique sectors. And yes, I have a lot of questions, and I've never seen answers. When studying microbes, it's important to remember that evolution and selection happen, even in the laboratory. The hand of Darwin is on all that lives. Nature is variable, and these images underscore that point. I recently had an email from a listener asking for more podcast content about fermented foods, that is, foods that are the product of microbial fermentations. And these include, what are not restricted to, cheeses, yogurt, kefir, pickles, sauerkraut, kimchi, and many others. In each case, there exists a whole community of microbes, often working together or in succession, that create that tasty end product. Thus, a tasty ecology. And so for that reason, I am beyond delighted to welcome Ben Wolfe of Tufts University to the podcast today to discuss this and related topics. Welcome to the Quality Quorum, Ben.
Hi, Mark. Thank you so much for inviting me. It's so great to be here.
Well, it is certainly a topic that lots of people are interested in. How would you like to begin? Would you like to pick a particular thing that the listeners and viewers need to know about with regard to fermented food?
Yeah, maybe a little bit about my journey into fermented foods, right? How did I get to studying things like cheese and sourdough and kimchi? I was trained as a fungal biologist, so my background is really primarily in mycology and all the really interesting things that fungi do. And as I was wrapping up my PhD on the really toxic fungus Amanita phalloides, the death cap mushroom, I was interested in doing something a little bit more edible, a little bit more relatable, perhaps, to a larger audience of folks that think about food. And I've always loved food. I'm married to a former chef who worked in restaurants for a long period of time. And so food has really been part of my life. And then Rachel Dutton, who I know you've had on the podcast before, was working on setting up this new lab to start to study cheese and cheese rinds. And Rachel was a bacteriologist. My training was in fungi, and cheese is the marriage of bacteria and fungi together in this delicious microbial community. And so it became this wonderful collaboration. She brought me on the team to help develop the system. And the rest is sort of history. And since then, I've been studying cheese and all these other fermented foods that you mentioned earlier.
You know, it's really funny, and I believe you're the one who sent me the protocol. Several years ago, I isolated my Micronauts, and I isolated some cheese-associated microbes. And I need to tell you, and this is coming from me, those are some smells that I was not prepared for.
Yes, they smell really intensely.
And what was interesting to me, Ben, is that I got the actual cheese aroma, but then there was this underlie of bacterial metabolism that I was used to. So it was very unusual.
Yeah. Were you using our PCAMS medium, or were you using something like LB, or do you remember?
I used both at one point or another. And yes, they get, I had students say, will you please promise you'll never ask us to do this again? And then I said, it depends on how you do on the next exam.
You're threatening your students with the cheese microbes. That's funny. In my lab, we study cheese, we study sourdough. We have some fermented vegetable projects going on right now. And the cheese folks always complain that they smell the worst because what's happening is those microbes, they're various actinobacteria, especially in that system. As they use various components of milk, which is casein, primarily a big protein, they chop it up. And as they do that chopping up, they release some sulfur compounds, things that kind of smell like cooked cabbage or farts, depending on how you like to describe it. And they are potent. They are incredibly potent. And then you also mix in some of the good staph species that you can find on cheese as well that kind of have like a sweaty, cheesy foot odor. And that whole combination is definitely like a funky cheese. So I think it's beautiful. I think, it's microbial metabolism. It's beautiful. And I think a stinky cheese is beautiful, but it definitely is an acquired aroma.
I wanted to ask a question about that. I had read recently, and if I can find this, I'll put it in the show notes. The compound geosmin made by a lot of actinobacteria, it smells like dirt. I used to have students sniff a plate, and then they would say, that smells like dirt. And I would say, no, dirt smells like streptomyces, because that's the truth of it. It's the geosmin, but apparently that aroma attracts springtails, these teeny insects, or arthropods anyway, and then they carry spores elsewhere. So whenever I see something odoriferous, I wonder about that. Is there any evidence of that with cheeses, that maybe this is, because I know there are some cheeses, I'm sorry, folks, if this disturbs you, that involve certain small other animals?
Yeah, it's a great question. So we were really interested in this in general because there is work showing that the volatile compounds, those aromatic compounds that microbes can produce, can sometimes be ways that microbes communicate with one another. And I've been working in cheese for a while, and every time I walk into a cheese cave, I was thinking to myself, am I walking into a microbial conversation? Are all these volatiles both interesting from the sensory perspective from cheese making, but also something to do with microbial interactions? And so I had an amazing PhD student, Casey Cusetta, who's now gone off to work in industry, who addressed that question for us. So she did these really excellent experiments where she would take microbes, so specifically the fungi that grow in our system that produce some of these funky aromas, and would grow them near bacteria, but they weren't touching. The only way that they could communicate was through these aromas. And she found that some bacteria, especially the proteobacteria in our system, respond very strongly to those fungal aromas. And specifically, we think they might even use some of those for growth, that they might be so carbon limited that any carbon they can grab from those aroma compounds can be useful for them to grow and survive in cheese. So that's one storyline where we've addressed those aromas. I think there's a lot more there. In cheese, you can often find cheese mites, these little creatures that crawl around on some of those long-aged cheddars and other cheeses. And whether they're using some of those bacterial aromas or fungal aromas to sense them and move around and be attracted to the cheese, it's an interesting question. I do believe that the fungi, people have shown that some fungal compounds are attractive to cheese mites. I'm not sure about the bacteria, though.
Oh, that's so interesting.
Yeah.
By the way, just in case people that are listening or watching are curious, you might want to explain what a cheese cave is, because first off, I think, hey, that's yummy, but that's not what you mean.
Right. Yeah. A cave made of cheese would be amazing. So the basics of cheese making, especially the cheeses that we study, we are not talking about plastic wrapped cheddar that you see at the supermarket. That's a great source of microbial fermentation. But in that case, you're just adding a group of bacteria called lactic acid bacteria to the milk, and they ferment the milk. It becomes a curd, and that's it, really. You don't get a lot of other microbial growth. You certainly don't get fungi. The cheeses we're talking about are things like Camembert, Brie, Limburger. These are cheeses that when you look at them have this layer of microbial growth on the surface. It's very obvious. It's either fuzzy or sticky, often pigmented. And that layer of growth develops during what's called affinage, or the aging part of cheese making. So you take that curd that you made in the cheese house, you put it into a cave, and those microbes then grow and flourish in that cave environment. And these caves can be anything from a big walk-in refrigerator to literally a cave in a mountain somewhere, or even just a concrete bunker. They're usually very humid, so 85 to 95% humidity. They're often cold, so typically, ideally around 12 to 15 degrees Celsius. And they're often dark, as most caves are. And those are the ideal environments that people have been aging cheeses like this for hundreds of years. And in the US, where you can see a lot of young, new American artisan cheesemakers, they recreate those environments through refrigeration or by building these facilities underground.
So I would presume there are populations of microbes that go back and forth between the cheeses as they ripen, as it were, and the walls of this cave.
Yeah. It's a really interesting question of what's living in the cave. We typically in my lab just focus on what's in the cheese.
Sure.
But there are several projects we've been doing that think a little bit more about the cave environment. One project a long time ago we never published, but was a fun sort of side project. We noticed there was this white fuzz growing on a cave up in Vermont at Jasper Hill Farm, where we do a lot of our work. They make amazing artisan cheeses.
They do.
And I'm sure you've had maybe their Bailey Hazen Blue or some other delicious cheeses. Yeah. And they're very microbially curious in ways that I absolutely love. So we were wandering around with one of the co-founders, Matteo Keeler, and noticed that their walls were covered in this white fuzz. And many of the cave walls, they're made out of concrete, had this. And what was interesting is, to me, it kind of looked fungal because it was fuzzy and white. But we put it under the microscope, and it was way too small to be fungal. The cells were really, really thin. It turned out it was an actinobacterium. And it's an actinobacterium that was closely related to, I believe it's either Nocardia or pseudonocardia, that can live on the stomachs of ants. These are leaf farming ants. Yes. That they use this this bacterium that produces antifungal compounds to fight off bad fungi in their fungal gardens. They create these gardens in tropical rainforests and other places to sort of grow food, and they need this one fungus. And they use a bacterium as sort of a biocontrol. And what I thought was really interesting is that same bacterium that's growing on the cave walls, is it sort of acting like the ant bacterium? Is there sort of a parallel there? We did some sequencing. We tried to do a little bit of chemistry. It was a side project. We never really sort of took it all the way to the finish line. But it was sort of thought of as beneficial by these cheesemakers that, having these good non-pathogenic microbes growing in this environment maybe can keep away bad fungi or keep away other bacteria that you don't want. So that's one way that we sort of explored the cave. Another really exciting project that we're working on right now, we've been working with Jasper Hill for over 10 years now. So we've actually been going back and sampling the same cave over and over and over again. And what's exciting about that is we can look at how cheese microbes are evolving over time. You were just talking earlier in the podcast about sort of phenotypic variation and how often, you domesticate things by accident in the lab. Cheesemakers were the original folks doing experimental evolution, sort of like Rich Lenski and other pioneers in microbiology. Cheesemakers have been putting out little petri dishes, wheels of cheese, in caves that have allowed microbial populations to grow over and over and over again. And they have domesticated microbes. And so what we've been doing is we've been going up to Jasper Hill and using time series samples we collected all the way back into 2016 to look at how have the phenotypes of the microbes there changed, but also how have the genomes changed. And it's a really exciting way to think about how humans are unintentionally, in this case, domesticating microbes in food systems.
It's something that you mentioned that I had said this in the early part. It's a big part of my brain pan here. This idea that what we do in the lab is a form of selection in so many different ways. And when you look at the history of microbiology, the first time someone sees, for example, bioluminescent bacteria, they'll find it's not stable on restreaking.
Yeah.
And so what they do is they tend to discard those and stick with the ones with a stable phenotype.
Yep.
Right?
Exactly.
And that was kind of incidental, but here you have a situation where it's truly directed toward a particular product.
Exactly.
And I just want to emphasize for listeners and viewers, what makes this type of microbial evolution so interesting is you can save samples along the way, as if we could magically keep different paths through evolution of vertebrates to watch how things change, which we don't have the ability to do, but we do with microbes.
Exactly. Yeah. And in our case, we're going back to a particular time point, 2016, where we were seeing this transition of this mold going from this very green, very fuzzy phenotype to quickly switching to become white. It's losing its pigment. And we're trying to figure out why. We're doing a bunch of sequencing and other experiments to figure out why. Fungal pigments, often blue, green, sometimes black or yellow, are melanin, and melanin can be important for fungi to protect them from the stress of light, from UV damage. And so it might make sense, and we see this in some other cheese fungi, it might make sense to let go of melanin production because you're living in a cave, right? You're no longer out in the stressful, bright environment of a meadow or wherever you happen to live in these sort of the surface layers of soil. You're in a cave where, sure, the workers come in and they tend to the cheese and they turn the lights on, but it's relatively dark compared to the stressful environment out in the world. So it's a really fun system for thinking about microbial evolution over human timescales. And it's not just in a petri dish, right? It's sort of out in the world, which I think is quite exciting.
And more to the point, so many times we have a fixation on pure culture and single colonies, neither of which exist in nature, to be truthful.
Right.
And yes, I know there are variations on that theme, but you know what I mean when I say it. But you're looking at community changes, and that's, I think, what is the real way to look at these things.
Absolutely. Yeah.
So in discussing the idea of the cheese caves having material on their walls that might in fact be vital to the proper ripening and development of the proper cheese, it kind of is reminiscent of the atmosphere in certain wine ripening. What's the expression, Ben?
Terroir is the idea, and the idea that maybe there's microbial terroir, that the taste of place is not just the climate or the variety of grape, but also the types of microbial communities you have in your environment.
Is this a similar concept, do you think?
There's a lot of conversation about this in the literature. There have been some reviews written, and for some foods, we can find unique biogeographic signatures. So we do find that different vineyards in the same valley can have unique microbial communities that might affect the properties of wine, or different producers of local yogurts might have unique microbial communities in the dairy and in the environment where they're making it that could affect the properties. But it's sort of a mixed bag. There are other ferments where we see no effect of geography at all. part of it depends on the level that you're looking at it. So are you looking at it at the species level? Are you looking at it at the very strain or genetic level? And I think part of it's too, it's just hard to get that resolution of sampling. So I think there is something there, and that strains matter, that unique types of species in fermentation matter, and those might be found in different places. But whether that is consistently spread across geography, and whether people should even be marketing the idea of microbial terroir, I would say that the jury's still out on that one as well. It's a fun, romantic idea. I love that microbes could be part of the taste of place. And we see variation in the microbiome. So whether it is structured consistently and predictably is something we're still figuring out.
And, I wanted to say that you sent me a copy of an article from Current Biology, which I'm going to make available to people. And I love this title, and maybe this would be a good place to jump off from. The title is Novel Fermentations Integrate Traditional Practice and Rational Design of Fermented Food Microbes. And this is interesting to me because if you look, for example, at the fermented product, and I'm sure I'm not saying this correctly, kefir.
That's great. Yeah.
Okay. Thank you. We make jokes about Kefir Sutherland all the time because we think that's funny, But the whole point is that occurred at some point in nature, and Lynn Margulis wrote about it, but it was due to human interactions. Same thing with sourdough bread and all the rest. But, nowadays you can start to be wiser about creating communities and modifying those communities to get particular products instead of what happened kind of randomly that got picked out.
Yeah. So we made this figure to try to explain that, and we can show that figure in the podcast. So there's a figure in the paper where we talk about how most fermented foods really developed as happy accidents, right? So there were a whole bunch of different food substrates around the world, a whole bunch of different microbes that could potentially grow on those food substrates. And people were just kind of making mistakes. They would essentially let something sit around for too long and a microbe would grow. Most of the time you get spoilage, but occasionally you get something that you smell it. Oh, that smells interesting. You eat it and you don't die. And so then you continue, the microbes continue and things move forward. And we don't really know because it's really hard to sort of have a fossil record of fermented foods. But we think through many years of sort of these accidental mixings of foods and microbes, we ended up with the diversity of fermented foods we have today. And it kind of looks like this big pile of spaghetti, and out the other end comes the sourdough, all the beautiful fermentations from koji in Asia and in China and Japan and other parts of the world. In Africa, there's a bunch of really interesting grain fermentations. In South America, you also have various grain fermentations. So all over the world, there are fermented foods that are largely from accident. And so what we're trying to argue in this new paper is that we can kind of take all the existing fermentations and now begin to think about, okay, let's not make it necessarily an accident. Let's start combining microbes and substrates in particular ways so that we maybe can come up with completely new fermented foods. And so another thing that we did in that paper, and we have another slide that can help us think about that, is we took all the fermented foods that we could get data on in terms of the substrate and the microbial community that lives in that fermented food. And we mapped it out in something called an ordination to kind of see how similar they were to each other. And we asked the question, are there gaps in that fermentation space? If we map all those foods out, do we see places where maybe new combinations of microbial communities and substrates, so foods, could we come up with new foods? And we found some gaps. Some of them might exist because if you start to mix those microbes and foods together, it might get a little weird. So for example, between like stinky tofu and delicious prosciutto di Parma, like that could potentially be weird. But maybe there's some interesting combinations to explore there. I found this paper really helpful too, because in addition to thinking about what is the current fermentation space and what are gaps, we also reviewed the literature and looked at what are chefs and what are scientists doing to invent in this space? What are the approaches that they're using? And there's some just really fascinating work happening. Everything from doing something really simple where you take the microbial community from one fermented food and put it onto a substrate that you wouldn't normally find it on, to taking the microbe from one fermented food, something like the microbe that's used to make soy sauce. It's called koji or Aspergillus oryzae. Some folks in Japan are taking that mold and sticking it on camembert cheese, where it never really would grow, and getting this sort of green, really wild-looking camembert that has very different flavor properties. And beginning to explore, what is that, right? What is that new thing? Is it actually new, or is it just camembert with a different coat? And all kinds of really interesting microbial ecology questions come up, but also cultural and food-related questions as well.
And when you use cultural in both senses of the term,
Absolutely.
which is awesome.
Absolutely.
Now this reminds me a little bit about some experiences I had with. I'm going to mispronounce it, kefir. Yeah. And you have roughly 20 different taxa of yeasts and lactic acid bacteria. They can't really be cultivated apart from one another.
Yeah.
And so this idea of an interdependent collection is fascinating to me, and how things enter or leave that association. And the reason I bring this up is that it had been written about. It wasn't original with me. When I take a standard milk-based kefir grain and I start to passage it on grape juice, just bear with me.
Yeah.
You get this fizzy grape drink, and the grains change their consistency.
Absolutely.
Now, I didn't have the ability to ask if the microbiome of those grains changed, but I'll bet they did.
Absolutely. Yeah. And so those kinds of experiments are interesting because it can give you fascinating new foods, but also maybe new microbial communities. And it seems like there's a lot happening. A lot of it's happening kind of in kitchens at home or in professional kitchens with chefs. And as in your experiment, we don't really have data to show exactly what's happening with the microbial community. So another thing we pointed out in this paper is let's start to work together, right? Often the microbiologists kind of work in their little ivory towers in academia, and the practitioners, sort of work off on their own, making all these fascinating foods, and sort of finding a space in the middle where we can provide more access to those chefs and fermenters to have tools like microbiome sequencing to understand what's happening under the hood. That might be a really fun space to work in. And that's something we've been trying to do, and others are doing around the world right now as well.
I have a question which is related, but we didn't discuss it before, and I want to throw it at you.
Yeah.
It's okay to say I don't want to talk about this. It's how red wine vinegar is made.
Ah, sure. Yeah, I know a little bit about red wine vinegar. Yeah.
So my understanding is there is not an inoculant for this. You simply have and I'm really sorry if this disturbs folks. I think it's great, but you know how this goes. Well, you have to know how this goes, because when you talk about microbiology, people look at you funny. So you have like a shallow dish, you pour the red wine vinegar in, and you get this biofilm on the surface. And those are the organisms that are using a lot of oxygen, and they're converting that ethanol that's in the wine into acetic acid.
Absolutely.
But no one's adding anything to it. It's just like when you make pickles. There are things that are naturally found on the surfaces of the leaves or in the air that we're talking about with vinegar. So I don't know how you control for that, although the idea of your cheese caves gives me some ideas.
Yeah, it's an interesting question, and those are called spontaneous fermentations. So in the world of fermented foods, you can kind of make them in two ways. One is through inoculation, and the inoculation can either be through an unknown community, like with the kefir grains, or like when you make kombucha, it's often a blob you just sort of stick in. Or you can buy pure strains and start to mix them together. So that's the inoculation approach. Or in spontaneous fermentations, things like chocolate, things like fermented vegetables, and some red wine vinegars, you just kind of take the food substrate and hope for the best. And if you build it, they will come, and you'll get the microbes that you should get. And the nice thing about spontaneous fermentation is that it generally works. It doesn't always, but it generally works. You generally get the microbes you want. And because it's spontaneous, you are capturing the microbial community of the place that you are in. And so you may be capturing something unique. We've done some work with fermented vegetables in the past, where we found that if you grow the exact same cabbage in different parts of Massachusetts, let them collect different microbial communities in those different parts of Massachusetts, then bring them back to the lab and ferment them, you get very different fermentations and very different microbial communities, simply because there are different populations and communities of microbes out there. So that's the beauty of the spontaneous fermentation. There is the risk, though, that if you are in one particular place and you don't have enough of those good bacteria that do the fermentation or the right mixture, you may get a funky ferment. And this happens sometimes with anyone that's tried to make sauerkraut or kimchi. Sometimes you go and buy a cabbage, you chop it up, put some salt in there, put it in a jar, and it just kind of rots. It doesn't actually ferment. That might be that you just don't have enough of those lactic acid bacteria that live on the leaves that can do the fermentation. But yeah, I think that's the thing about fermentation that's so fascinating is that it generally works, even though we're relying on this variable and sometimes rare population of fermentation bacteria.
And for some of these fermented food items, by the way, I'm loving this hashtag funky ferment. I think that's a band name, or it should be a lab name, right? There you go.
Yeah.
So in any event, some of these assemblages have been used by people for thousands and thousands of years. And again, as you mentioned, in many cases, they're not transferring anything. I hope you'll talk about sourdough starters in a little bit.
Absolutely.
But what we're getting to is this idea that they are just part of, and I still like if you build it, they will come, which goes back to the Bass-Becking idea of microbial ecology, that everything's everywhere. And
Absolutely.
I don't know if that's true, but I do think anytime that you're associating with plants anyway, you're going to tend to have certain members that would lend themselves to fermented material. I mean, one of the ways that most of us who teach microbiology teach a little bit of microbial ecology is to look at how about how sauerkraut or kimchi forms. And there are a series of successions that take place where the first inhabitant creates an environment that the second benefits from.
Yeah. So, part of the succession is first creating that low oxygen environment, and then moving through that low oxygen environment, so sort of shifting metabolism as you get less and less oxygen, and you have to use different parts of the plant that are available to you. And that's been really fun because it's a very consistent succession. But it is somewhat dependent on that initial microbial community, what you have there, the interactions that are unfolding in that microbial community. But yeah, I think that's the power of sauerkraut, that it almost always works. But, if you ever want to play around with this, try going to a farmer's market and getting a couple different types of cabbage, or try to get the same, just green cabbage from a couple different supermarkets, bring them home, kind of keep them separate from each other as you make the sauerkraut, and see how they ferment. You might notice that they taste different. You might notice that the pH changes. And part of that is probably because of the slight differences they have in microbial communities. Yeah.
I think this is so interesting. And I want to say parenthetically, and we will talk about this, you have a lovely website where you do outreach material.
Yeah.
Where there are instructions for people to make some of these items.
Yeah. So we run a website that I co-founded with Bronwyn Percival, who is a cheese buyer for Neal's Yard Dairy in the UK, called microbialfoods.org. So back before I started my lab, I saw the need to create a website that would explain the science of fermentation and sort of take all of this scientific information that was locked away in journals that you had to pay for and take it to the people. And it needs a little bit of love. It's been a while since we've written some new material, but there's a lot of back material on there about mostly cheese, but other kinds of fermented foods as well, to teach you about what you need to know about those foods. So check it out, microbialfoods.org. And we've got some really fun, infographics on salami microbiology, some really interesting cheese posters you can print up and hang up in your kitchen, perhaps. Yeah, I love that website.
No, and I think it is good because, and one of the things I like about this general topic, and again, I'm so glad you're here, is that everyone encounters fermented foods in one form or another.
That's true.
Sometimes in ways they hadn't expected. And that's why I find the whole subject fascinating. But often when I talk about microbiology, people have an automatic negative, I make fun of it right here, the term germ.
Yeah. Yep.
So when Pasteur used the word germ, he meant a piece of life. He was fighting with the people who were believers in spontaneous generation. So that's why I take this bottle of wheat germ and cross out the wheat, and then I put giant microbes in there. But
yeah, it's true that fermented foods are an amazing platform for outreach and education because it's a positive association that people have with microbes. And they may not know exactly what microbes are there, but what we've done in the past through lectures, we've had this little microbiome booth we took to farmers markets, and we'd have microscopes and all that there. Is we would first talk about the food and get people comfortable talking about their experience with the food, and then start talking about the microbes. So that fear factor that people have when they hear bacteria or fungus or yeast kind of diminishes, and they engage with the food first, and then you can start talking about the microbes. And it's worked wonderfully. You can very clearly show diversity in microbial functions by you having those plates we were talking about earlier. You have streaks of different bacteria and show that they smell just like the food. Under the microscope, you can show microbial communities in something like a fermented vegetable or sourdough. It's a really powerful system for outreach and education. And it's been fun to see a lot of other people across the US and the world also exploring how do we teach the public about microbes through fermented foods. And so, I mean, there's lots of platforms for doing that, but I think this is a really delicious and fun one.
You planned that one. I heard it. I was coming up there. I knew.
Yeah.
And you're also going to say that was a tasty idea as well. I'm getting there. Okay. Yeah. So, by the way, I noticed behind you, you have a giant poster, and it's kind of, I guess it's a hanging. I'm sorry.
Yeah.
And what I like about it, and for listeners, what we're looking at are looks like certain types of fungi that are growing up. And that's a harken back to how you started in the field and all the rest.
Absolutely.
And it's a nice reminder.
Yeah. I mean, I think in general, bacteria get a lot of attention in the world of microbiology, which is great. They're wonderful. They're very important. They're incredibly diverse. But one thing that my lab has been thinking a lot about is the fact that often where you find bacteria, you also have fungi. In soils, even in the human gut microbiome, we have some fungi that are there. And so thinking about how do fungi and bacteria interact with one another is a very common theme in our lab and other labs as well. And I think this has been growing in part because the barriers between fields like mycology or fungal biology and bacteriology have been dissolving, and we've been all talking to each other more and going to meetings and sort of coming together under the umbrella of microbial ecology. Mm-hmm. And so we've been finding that fungi, especially in cheese, are incredibly important. They're sort of a keystone species for cheeses and cheese rind microbiome. So they build physical networks that bacteria can grow and move on. They are the decomposers, just like in the forest. And that decomposition is releasing nutrients like free amino acids. They are also very important producers of metabolites, secondary metabolites that can benefit themselves, but also can be really beneficial for neighboring bacteria. So you really can't think about the world of microbes in most microbiomes by ignoring those fungi or any other group like protists, viruses, etc. One thing that we do in my lab is help cheesemakers when things don't go as they have planned. This doesn't usually happen very often, but sometimes weird things pop up. The cheese rind suddenly changes. And one particular case, we found that a fungus, a penicillium, from the group that brought us penicillin, started to make this really clear zone of inhibition. So this area where we could see it was killing bacteria. And the cheesemaker called us up and said, yeah, my rind is disappearing. Like all the good bacteria that are normally there are disappearing. And that's when this fungus showed up. And the reason that that fungus is antibacterial, we went on to find, is it makes a compound called serotin, which is a secondary metabolite. It is known to inhibit the growth of insects. So maybe the fungus uses it out in nature to keep itself from being munched on by those springtails. But it turns out it's also very antibacterial. And we found that, it inhibited both proteobacteria, actinobacteria, firmicutes, all kinds of groups of bacteria. And what I think is really exciting, we worked with some collaborators at the University of Wisconsin-Madison, Nancy Keller's lab. We can go in and knock out the gene for serotin to actually show the exact role that it plays in driving this bacterial community. And for me, it was striking to see that one single chemical produced by this fungus can have such a dramatic effect on the neighboring bacteria that are living with it. And I think this is happening all the time in bacterial fungal interactions. We just haven't figured out the chemistry yet. And that is, again, all basic biology from a cheese rind, which is really exciting.
Can you spell serotin for me?
Yeah, it is P-S-E-U-R-O-T-I-N, serotin.
Wow.
Yeah, it's a relatively understudied chemical group. There are different flavors of serotyns, and one thing we did in the paper was just sort of check a whole bunch of fungal genomes to see what other fungi are making serotyns. But for me, it was also just so striking. We often see zones of inhibition when we're looking at antibiotics and how they affect the growth of bacteria, but a fungus can make a huge zone of inhibition in the cheese rind system. And again, this was super important for the cheesemaker. It was a problem for this particular cheesemaker.
It's interesting. One of the things I'm having my students do is look at isolates from the soil, from lichens, and from mosses to see if any of the microbes there are making antimicrobial compounds.
Cool.
We always find some. Now, are they important? That's what Joe Handelsman's Tiny Earth program is all about.
Yeah.
But it's all there. And I do want to say something about this, that there is a school of thought that says, kind of the Margulian view, that everything always gets along, versus a more Marshall view where everything's fighting. And as always, extremes are not where the truth lies.
Yeah.
I do think that microbes compete for real estate.
Absolutely.
They also can cooperate, of course.
Absolutely.
And isn't it funny to think about both of those things taking place on a cheese rind?
And at the same time, I love that you bring up this point. We often think about either like the costs or benefits of living with a partner, whether in life or as microbes. And what's interesting with these fungi is they can be secreting both beneficial and antagonistic things at the same time. So they can be, for example, secreting a protease that's chopping up that cheese substrate and releasing free amino acids, which are good for the neighboring bacteria, while also hitting it with an antibiotic. And so what the bacteria have to do is figure out, what am I going to do with that, right? How do I balance those costs and benefits? And so typically when we study microbial interactions, we just study one at a time and think about, just the positive or negative. But it's this whole mishmash of all kinds of metabolites and ways they're interacting. And it's complicated, but I think really exciting.
There are a lot of people who don't like the term omics because it becomes a buzz term. But I think using the term metabolomics makes sense, because you have such a universe, especially small molecules, where people haven't really studied them. And they do definitely have an effect, both locally and distant, in any type of community. Again, because we tend to focus on Petri dishes and, I show my age, 2059 tubes, right? And that's not how any of these things grow. They do grow in association. And it's really interesting to think about, and what a wonderful way to use a wheel of cheese to look at those things.
Absolutely. It's a relatively simple system with not that many species, and we can do things like genetics in some of the organisms. And a shout out to Laura Sanchez, a chemist that we worked with at UC Santa Cruz, that's helped us a lot with identifying these compounds. Like, we can't really do that ourselves. We're not chemists. And so it's folks like Laura that like to hang out with and sort of play with the microbiologists to understand the chemistry side. And we've found lots of different examples of, the metabolites aren't turned on unless they're interacting on the cheese. It's been a really fun way to get to mechanism in the system. Yeah.
So could we talk a little bit about sourdough?
Yeah, I just pulled up the image I wanted to talk about. And, it's such a important fermented food that obviously during the pandemic really became popular as we had yeast shortages and people were just trying to figure out ways to de-stress. So we started working on it long before the pandemic, actually, long before it became really popular. Rob Dunn at NC State University approached me and said, I want to study a fermented food with you, Ben. And I want to he has this amazing power of getting community scientists. We used to call them citizen scientists, but I think community science is a much better term to include everyone, to work with scientists, academic scientists, to sort of get a whole bunch of samples and ask a question. And sourdough is a perfect system to do that because there's so many people making it all over the world. So we put a call out. We ended up getting over 500 samples of sourdough starter sent to my lab at Tufts through the power of Rob's network. There's a great picture on one of the slides of an undergrad, Stravia, who used to work in my lab, that was helping Liz Landis, the PhD student in my lab, work on this. They just kept getting envelopes and envelopes delivered, mostly from the US. And the thing we wanted to do was figure out, what's living in sourdough. And at the time, surprisingly, two things were sort of true. One is that most sourdough research was done in Europe, where sourdough has been made for a long period of time. It's super important in French, Italian culture. And so many studies were focused just in Europe. And the second thing that was interesting was that many studies were still culture dependent. So people were really relying on plating stuff out to figure out what was there. Makes sense because you can actually grow many things from sourdough, but we wanted to see what these culture independent sequencing approaches could tell us. And so on another slide, I have a big picture of what we learned from the omics, the sequencing that we did. We have data on the top of that slide. There's sort of a top row for the bacteria. And in the sort of second row of colorful bars are the data for the fungi that we found. And what's great about having over 500 samples is you can start to ask questions about, do you see consistent types of communities forming in sourdough? Maybe it's just all kind of random and everyone has very different communities. Or do we see reproducible types forming over and over again? Other things you can start to ask are, do you see associations between particular microbes? Do you see 2 or 3 species always co-occurring? And maybe that hints at some kind of interaction. And then another really important thing for sourdough that we can start to think about with these large data sets are questions about geography, right? Often people talk about San Francisco sourdough, or people talk about their sourdough in one place having a unique flavor, at least, but does it have a unique microbial community? And one thing that we found in the sequencing data set is, one, that the yeasts were dominated by one thing, Saccharomyces cerevisiae, the bread or beer yeast. That makes sense. It loves bread. But there were some really funky yeasts out there, like Wickerhammermyces anomalus. It smells like bananas. It's amazing. I should send some to you because your students will love that. It's such a funky and interesting and beautiful, actually, on a plate too. You'd love it. I'll send some in the mail next week to you, I promise.
Wonderful.
And then the bacterial communities were much more diverse and much more variable. But we did also see reproducible communities. So we saw very distinct types of communities forming over and over again. Part of that was due to people buying it from companies. So for example, we could see a signature of the King Arthur sourdough starter in our data set because people reported buying it from the King Arthur Flour Company in the United States, in Vermont. And then the other thing we found that was kind of a little bit of a bummer for some folks was that there was no signal of geographic structure in our data set. So when you're looking at that big diversity of sourdoughs, one chunk that kind of all looks the same didn't come from one particular place. And at first we were kind of disappointed by this as well, because we were thinking that there would be some geographic signal. But when you think about it, we were talking about this earlier, fermented foods are shared and moved all over the world. People are sharing cultures, they're moving with their cultures, they're buying them online. And so that geographic signal would get completely swamped out by that human migration, that humans dispersal. And so, sorry, San Francisco, there's nothing special about your sourdough microbes. Your sourdough is great and wonderful and amazing for other reasons, but there are no, as far as we know, there are no unique communities of microbes in the Bay Area that make it special sourdough.
This reminds me a little bit of microbiome work. And I want to forgive you. I want you to forgive me for what I'm going to say now. I like to think about things that have co-evolved, say, with the gut, and you have things that pass through. And then the question is, can any of them become a temporary resident, a squatter, if you would? That's kind of a pun, I guess.
Right.
And then sometimes they become residents. And that process by which you're passing through or become a resident is a really interesting one to me, because that had to have happened. So if you think about all these sourdough starters, they're not being kept under super sterile conditions. Absolutely. There are going to be all these passerbys, and some of them are just going to be lost, and others are going to be able to persist there. And it's a really interesting question, and it's very different than the way we do things, generally speaking, in a laboratory.
Yeah, it's sort of a question of invasion and sort of when are microbes successful at invading. And an interesting anecdote to that is from the sourdough data set, the King Arthur sourdough starters. We had quite a few of them in our data set. They have one bacterium and one yeast, so very simple community. And we had, I think, over 20 participants that reported working with those sourdough starters. And interestingly, they were identical, right? So after having been in different kitchens, as you were saying, that are very messy and very open, we found that the community stayed the same across all those different participants. It was exactly the same as the King Arthur starter that we bought and sequenced. That was really surprising to me. So I have some folks in my lab right now that are trying to invade it to see if we can add in those new microbes and do they persist. And surprisingly, we're finding that they can actually. So it could again be human. So maybe when you have the King Arthur starter, it's a wonderful, beautiful thing. But then you get an invasion and you don't like it and you throw it away and you start again. So maybe human selection actually weeds out anything that deviates from that original King Arthur starter. We're not really sure. We're still working on that. But it's a really great question about thinking about when do you get invasion? When is it successful? When is it not? Yeah.
It's very interesting because Rob Dunn did always inspirational to me. I haven't spoken with him in many years, but I so appreciated and you probably remember this, the belly button microbiome. Yeah. This is how this started.
Yes.
And this was actually my inspiration for looking at things in water bottles from students.
Yeah.
And you think you're selecting for biofilm formers and things of that nature. In plastic water bottles, I tend to see a lot of methylobacter, and I think, I can't prove this, but I think that they're living off stuff being leached out from the plastic.
Oh, interesting.
Because I don't see them in steel bottles. But what I'm unable to do is and again, I really appreciate what you said about community versus citizen science, but sometimes because of my age, I will use old-fashioned terminology like that.
Yeah.
So I would love to have a situation where I had funding to do this in a large group.
Yeah.
And I would love to be able to look at deep sequencing from each of them. I don't have those facilities, but I see certain keystone organisms every time I look in a water bottle. And that means something.
Interesting. Yeah. And it would be a perfect project for one of these large scale working with the public. Because, I mean, just come to my college campus. There's always water bottles lying around the lost and found. We could do it. We could definitely do it. Yeah.
And it's so funny because then when students do the swabs, and I'm all about staying away from, and I don't know if you've heard me say this, but I have this bugaboo in my head that we believe in devil microbes like we believe in angel microbes. That is to say, they're always bad or they're always good.
Right.
Both are wrong. It's mostly meh. Mostly They don't care.
Right.
But and then I also talk about swab stories where we actually look, there are microbes in this environment. Well, of course there are. So when I have the students swab their water bottles as a first step, they're getting grossed out. So I'm defeating my own purpose sometimes.
Yeah. Yeah. I mean, this idea that microbes are everywhere. I guess give them some sourdough, maybe distract them with that.
Yeah. No, I love that idea. I think it's wonderful. And like you said, you're asking good questions. You have facilities and you have equipment to study those things. And it's really wonderful.
Yeah. And I love this idea that we're finally starting to decode the microbiome of the places where we live. We don't have to go to faraway tropical rainforests or deep under the ocean, which are great systems to study. I'm not trying to downplay the importance of the systems, but the world that we live with and interact with every day is also a microbial community, and we should figure out what's there, including the foods that are there.
Ben, I used to work in a biotech company back when dinosaurs ruled the earth, and we were looking at bacteria that made biological polymers that were important for industries, as xanthan gum is, which is made from Xanthomonas campestris.
Yeah.
It's that exopolysaccharide used as a suspending agent, and we had 2 proprietary organisms, and they did not send me, but they sent other people at my company all over the world to collect soil and water samples. But these 2 moneymakers were found in northern San Diego.
Huh.
So again, there's the old poem, you find a universe in a grain of sand.
Yes.
That's the microbial world.
Absolutely.
To misquote William Blake.
And it clearly continues to fascinate both of us, right? And I really appreciate all the things you do online and through this podcast and at meetings to sort of keep us all stimulated and keep your students stimulated, but the entire world stimulated by thinking about what is out there and what does it mean that it's out there, and asking these really important questions.
And that's why I made up this OMG for, overwhelming microbial greatness, and WTM for what the microbe. Because these are things that we talk about all the time. I mean, pink pigmented facultative methylotrophs that live on the underside of leaves, all these different things are wonderful.
Absolutely.
So this is going to sound perhaps a little bit trivial, but there's an image in my head in Italy where people are stomping on grapes as the process of winemaking. So this gets me thinking a little bit about the transference of microbes, not necessarily from the feet, being kind of a way that you're inoculating a fermentation. Do you have thoughts on that?
Yeah, it's a question I get often. There's an idea of in Korea of kimchi hands, that certain people have different bacteria on their hands that might contribute to the fermentation. There's also an idea in sourdough that there could be movement between humans and fermented foods. We actually, I would argue, don't have great scientific evidence to show that any human-associated strains are directly contributing to fermented food microbiomes and the way that they ferment. I am not aware. There's some anecdotal things, and there's some papers out there that are very preliminary that suggest connections between humans and the fermented food microbiome. But often it could just be that if a human's hanging out a lot with a fermented food, like, for example, working with a sourdough starter a lot, they may get that starter on their hands. And then when you sequence their hands, you just pick up sourdough microbes because they're around sourdough, not because they are hosting those microbes. It makes a lot of sense because, fermented food microbes have evolved to live on plant leaves or in wet dough or on a rind of cheese. And so they don't necessarily have the right equipment to deal with the human body. But
this is very arid.
Yes.
It's very salty.
Absolutely.
And we forget.
I wouldn't be surprised if eventually someone does find some connections, but so far I'm not aware of any really great links directly between the two.
I'm watching the time carefully, and I do note that there's one thing that you do that we haven't talked about, and I'd like to, if that's okay.
Absolutely.
You have done some outreach helping people understand about their gut microbiome.
Right. Yeah. Yeah. So I was often getting questions from either students or from the public when I would talk to them about, what if I want to understand my own gut microbiome? I don't work on human microbiomes. I've collaborated with folks in the past, and I'm always interested about, what do you eat and how does it affect your gut microbiome? And that's a whole other conversation. But I decided, okay, well, one way I can explore this is to think about what are these home microbiome test kits that are available. As someone who uses microbiome sequencing data all the time and thinks about how you get that data, how are these companies potentially generating it? And then also, what are they telling you, right? What are they telling you about your gut? And I did this sort of as a fun, just personal experiment, but also I wanted to write about it. And so I wrote about it for this great sort of open resource called The Conversation, where academics can write about research. And so I got kits from 3 different companies, not all of them that are out there, just 3 that I thought of. And from the same sample, I took a little bit of sample and I sent it to each of those 3 companies. And then I said, what do you find? Tell me about my microbiome. And it was really fascinating to see that I got very different answers from each of those different companies. And maybe it's not surprising, right? So they're all using different sequencing techniques. Some are using RNA-based, so they're looking at active. Some of them are using DNA-based, so they're looking just at DNA. So you're going to get a very different answer that way. But also the way that they interpret the information, the way that they use databases to suggest things about the health of your microbiome is also fascinating. So, on one of the images that I provided, there's this sort of output that I got about my microbiome that said that my gut score was 5 out of 10. So not great. I was good in the diversity area, so I had an 8 out of 10, so a B-ish. But in terms of phylobalance and some of these other metrics they had, they were not great. But other companies said I was okay. So it depended on the company, it depended on the information. At the end of the day, I actually did learn something about my gut microbiome. I found that I, have this one microbe that maybe one day, I wrote about this in the article, like a long time from now, could potentially pose a risk to me. Maybe, we'll see. It's never clear from these kinds of data. I also learned a little bit about, just the overall diversity I have relative to an average human gut microbiome. But I also learned that these companies largely are collecting this data to try to sell you other stuff, right? They're trying to sell you supplements and probiotics or nutritional advice, which is sort of not surprising, right? They're not just doing this to get the information to you. But I do think if anyone is interested, it's a fascinating way to sort of look at the microbial community you're carrying with you, and even if if you have a change. So, for example, I collected this data before I had my appendix removed, where I had to take a whole bunch of antibiotics, and I did it again after with the same company to see if it changed. It didn't, surprisingly for me. My microbiome was almost exactly the same. So it's useful too to think about, as you go through different changes, what can your gut microbiome do? How does it change? Whether or not this is information that eventually doctors will be collecting from all of us and becoming part of our overall way of thinking about managing our microbiomes, I'm not really sure. But I had a really fun time sort of exploring this topic and thinking about what it means. And I've heard from a lot of people too that they found it useful for thinking about both the benefits, but also the big limitations of these home test kits.
Just as I mentioned this idea of angel microbes versus devil microbes, we can be too flippant about issues. We all see videos and animations of the microbial world that are a little bit much.
Yeah.
Or we can be too dry.
Yeah.
And what I'm interested in is being able to treat the public as if they can understand things, because they can.
Absolutely.
To talk to them, not at them, and certainly never down to them.
Absolutely.
And that's one of the things I like about the guests that I have on this podcast, is we can address issues like that for interested people in a way that's supportive, I think. Absolutely. So absolutely, we're going to put a link to that one, because I've read your article. And other things about what we've talked about today in the show notes, and I hope people will look at them.
Perfect.
So adjacent to the idea of angel microbes, there's a lot of information the public sees touting the benefits of fermented foods in terms of health. Now, certainly there's a lot of evidence that some of the digested material, the fermented material, have a prebiotic effect, certainly. But is there any evidence for more direct medical benefits?
It's a fascinating question that I spend quite a bit of time thinking about with a lot of other fermented food researchers a couple of years ago. And I can send you a link to that paper to have in the show notes as well. We reviewed the evidence for whether fermented food microbes can have probiotic effects on humans. And that's directly that the microbe itself, the microbial community, is doing something with the human body. And to be honest, it's pretty limited, in part because it's remarkably hard to do controlled clinical trials where you can get people to just eat certain foods or not eat certain foods. There is some evidence with some fermented dairy products like yogurt, which has been very well studied, that there can be some benefits. But often, as we were talking about earlier, the microbes are transiently passing through you, and it's their metabolism as they're passing through that can have an effect, not necessarily the fact that they then sort of colonize your gut and become part of your gut microbiome. So there's very little evidence. If you turn around in the back of a bottle of kombucha, it seems like it's promising the world in terms of probiotic benefits. But honestly, there's actually quite limited evidence. The evidence for nutritional benefits, right? The idea that when you make sauerkraut, that those bacterial communities are breaking down cabbage, the leaves of cabbage, and producing metabolites that might be beneficial for you. Vitamin K, for example, that is very clear, right? And that is because of the microbial activity. The direct probiotic benefits, the idea that the microbes are changing your immune system or, colonizing you, that is still very preliminary or completely lacking for some foods. So stay tuned. I think we're going to learn more as we get more opportunities to dig in. I think there will be some fascinating crosstalks between the microbes and the human microbiome, but it's still in the early days for that.
Do you have any last minute advice for people or things you'd like to say?
I think that people should just be really curious about what's living in fermented foods. Try new fermented foods. I'm trying them myself all the time. Be microbially curious about the world in general, but specifically about the food. And yeah, I think microbes are so fascinating, and we just know a very tiny bit about all the things they're doing. And so, if you're at all interested in this topic, there are lots of great labs all over the world, starting or have been around for a while, that have PhD students they're looking for, that have outreach websites that they're doing, or various other things that are educational activities. Check them out, because the food microbe space is a really fun and interesting space to learn about microbes.
Absolutely. I couldn't agree more. Final thought for you. I did this when I spoke with Rachel Dutton. I'm going to do it for you. Are you familiar with the author Frank Herbert?
Yes, somewhat.
Now, he wrote the novel Dune.
Exactly, yes.
He wrote many other things,
yes.
And he wrote a novel, and please forgive me, during his drug curious period, because that's what it was, called The Santaroga Barrier. And the whole point of the entire novel, Ben, is that there's a town where the people are in symbiosis with some kind of fungus that's in their cheese caves.
What?
And it's opened their minds. They've created a group mind. I am sending you a copy.
Yes, I need to read this. I have not read the Dune novels. I found the movies to be entertaining. I have a Dune lover at home, so I've learned a lot about Dune. So I need to... what's the name of it again?
You don't have to worry. I'm sending it to you.
I'll send you the yeast. You send me the book.
and by the way, I do put together a packet for people who have been on the program, and it will include a button that I know you'll appreciate it, easily distracted by microbes.
Absolutely. 100%.
Well, I'd like to thank you once again for being on. This has been a wonderful episode. I think I've learned a lot, and I hope our listeners and viewers have as well.
Thank you so much, Mark. This has been great. Thank you.
Thanks so much. I really appreciate it. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, and comments to me at mattersmicrobial@gmail.com. Show notes, and tasty show notes at that, from today's episodes can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Ben Wolfe is an associate professor in the Department of Biology at Tufts University in Boston. Many, many thanks, as usual, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile each time. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.